The next person who's going to talk is Yana Bromberg. Yana is the leader of theme 2, which is a bioinformatics group. It's an attempt to understand the evolution and origin of the structures and functions of metabolism. Yana got her undergraduate degree in computer science and biology at Stony Brook University. She did her PhD at Columbia University and a postdoc there. And then she was recruited. I recruited her, frankly, to Rutgers in the Department of Biochemistry and Microbiology, but she has interests across many departments and schools within Rutgers University. So without further ado, Yana, you're up.
So the original title for this presentation was Metallic Signatures of Life, but for some reason, the PowerPoint decided to change it to Microbial, or maybe I did at some point, but it was Metallic Signatures of Life. All right. So this is shamelessly stolen from Shauna Morrison and Vic Nanda, but this is the motivation for the things that I do. So if you think about Earth about 4.5 billion years ago and Earth as seen recently, there is a big difference. And that difference is due to life appearing on the planet. And I think the motivation for me and for the work that I do for ENIGMA is really about how does life interact with the environment that it is in order to make it more favorable to itself and for us as well. All right, so this is just a brief reminder for those of you who were not there when we first applied for the ENIGMA project. There are 3 themes here, and Vic had talked about the theme 1 effort. I lead the theme 2 effort, and we've had 2 postdocs over the years, Adrian Horfrost that Vic had mentioned as well, and Ariel Aptekman, both of who Vic had mentioned before, but I'm going to talk in more detail about the stuff that they had done. And I'm sure Nathan will talk more about theme 3, so I'm not going to focus on those themes, but given my location in this arrangement, right in the middle there, I sort of dip my hands into both directions. So the first thing that I want you to note is that there is a lot of machine learning arrows on this graph and this graphic. And the hope is to eventually do a lot of the machine learning that we proposed to do. But in order to do machine learning, you need to collect all the data that will allow you to do this, right? Data is not immediately available, and collecting this data in all the ways that we can, by both collecting it from experimental databases and producing it using other subcomponent of machine learning methods, is one of the most important things to do for us in theme 2. Okay, so let me start. There's 3 things that I'd like to talk about. The first one is summarizing the relationship of protein structure and metal binding. Now I'm going to go really fast through this because those of you that have been around these talks the past couple of years have seen all of these slides before. So this is a nitrogenous. The color scheme here is predicted, right? So this is a computational method for recognizing functionally important residues in the protein structure. The things that are residues that are highlighted in red are important. The residues that are highlighted in blue are not very important to the functionality. And the ligand in the middle is an iron sulfur cluster. And the one thing that you see very clearly there is that the residues that are important to the functionality of the protein seem to be very close to the ligand. In fact, Stefan Sen, that had worked on this project early on, this is even before the ENIGMA project got started, had said that the sphere in the protein structure of radius, he talked about 18 angstroms. There had been studies since that said 15 angstroms. But the sphere around centered at the ligand of the protein structure includes most of the functionally important residues. And this is the assumption that we went ahead with. The idea was further that if you wanted to align 2 protein structures in order to figure out whether they're functionally similar or not, you would take those important residues, the structure of those important residues in that sphere, and structurally superimpose it on another sphere, right? So you can compare two protein structures of functionally important residues and try to infer if they were similar or not, because that would allow you to look for similarities across the structural regions overall. Okay, so what Stefan had shown is that in order for this alignment to be meaningful, and I don't know how many of you look at these all the time, but the idea here is that there are two proteins, one is blue and orange, and another one is green and red. And only the piece that is in red and orange is the overlapping of these two proteins, right? So if you look at this overlap, how do you know that this is a meaningful overlap? And what Stefan had said is that actually you can infer that on the basis of how close the ligand centers are. So if the functional residues are the ones that you're looking to align, it's important to see how they carry out the function that you're considering. So these ligands that are together should be overlapping, right? So he had come up with this idea that ligand centers need to be overlapping. We had converted that idea over time into an optimization of sequence alignment from structural alignment that would allow us to compare any two structures in the PDV that bind any ligand, right? And we can score that. And if we do that, we can build this network. Now, the idea here is that every metal binding substructure is a node, and the edge is the connection, that this score of how similar they are that we have developed using these sequence-based alignments of the structural alignments, right? So if this is too complicated, I'm sorry, I don't really have too much time to explain it, but there is a paper now out, and you can feel free to ask me afterwards how we did this. Now, if you look at that network very carefully, what you see is that there is a bunch of clusters. So the cluster on the right in orange, this is the copper binding proteins. The pink clusters on the left, those are the heme binding proteins. And what you see is that, first of all, things that bind the same structure tend to be more similar to each other. But the second thing that you see is that everything is connected. So this network connects 99% of the PDB of all kinds of different ligands. So that suggests that either there is some physics limitation, which requires that all protein structures that bind a metal ligand are similar, or they have had a very small number of origins from which they had diversified into what we're looking at, right? So that's the main takeaway from this particular network. Now, if we analyze this network in more detail, and I'm going to skip the detail, but I'm going to tell you that what we're going to get is central spheres, that is spheres that look most, oh, I apologize, before I get to the central spheres. So the one thing that is important to note is that different substructures look more similar to each other than others. You saw that all copper binding structures looked similar. But also, if you were looking at the similarity of copper binding versus iron binding structures, you would see that copper binding structures are more different from those structures that bind other metals. And this is what we thought was in line, was the availability of the metal. Now, Nathan tells me that's wrong, but we thought that was in line with the availability of the metals in the primordial soup, and therefore evolution of particular structures to bind particular metals. So you see that square in the middle of dark blue, this is the ions, the structures that bind ions that are similar, and the stuff in light blue all around suggests that there is less similarity of these structures. Okay, so back to our story about the central spheres, which I find exceedingly exciting. So if we reduce this network, the one that I had shown you before, what you could see is that there are central spheres here. And if you think about what the trees are, they're just splayed out networks. So evolutionary trees are splayed out networks. And you can pull basically up this network by the middle over there, suggesting that these central spheres look very much like ancestral proteins. Interestingly, the vast majority of these central spheres that we found are oxidoreductases, which is in line with our assumptions about early proteins. Another thing that we found is using Diego Ferreira's, our collaborators' work, is the Lego building blocks of these central spheres. I'm borrowing Vic's phraseology here. And what we saw is that there were 31 or so of these building blocks that could be formed, clustered into these 6 more or less similar clusters. Now, what was interesting about these Lego blocks is they were found in large quantities around all proteins, not just metal binding proteins in the PDB. And what we tried to do is figure out when they would have appeared, time-wise, when they would have appeared. We used Caetano Anolis's work on identifying the ages of the scope folds. And then we mapped each of the tiles. There are, remember, 31 tiles that we mapped to these scope folds. And we said, okay, how does it look in terms of age? So lower down means earlier ages, so 3.8 billion years ago or so. That was the earliest P-loop that Caetano Anolis finds. And that is C37 over there in the bottom right-hand corner. And that one has 27 tiles out of the 31 tiles that we had. Now, that means that in evolutionary history, going up this thing, we're seeing fewer tiles, fewer distinct tiles being used by the sculpt folds, up on the way, all the way to about 0.75, which is about 800 million years ago or so, right? And then we get this more folds again. And so to me, this looks kind of like this. So you got this Lego slum on the bottom over there. We're using all kinds of tiles, right, to build something. And then as you go up, you get better at reusing things and making more compact representations, right? But then something happens and we get, again, this Lego slum up there. And the idea, this is recent and I have to go through this, but the idea that has occurred to me that this is about the time that the animals appear and the early plants appear, but also this is when we get a slight bump in the oxygen concentration. So maybe that has something to do with that. So re-optimization of the existing tiles to something that we knew that we're doing. So that's the first step that I wanted to talk about. The second thing that we did was look at microbial function, so catalytic activity and the environment. And to do this, originally we had this method called MyPhaser that worked by alignments. So basically you take the known reference genes from known assembled microbial genomes, you get these reads out of the microbiome sequencing and you align them to the genes that you know, and you assign function in this way. This is the way that normal assignment of molecular functionality happens in everyday life of microbial biologists, right? So this is how we do it. We thought that we could do better, but I want to show you first that even that way of assigning functionality actually relates to the environment in which the microbiome resides. So we have the deep horizon oil spill map. And we have microbiomes whose functions we can annotate just using alignments, that just the way that I had shown you before. So every microbiome can be represented as a vector of functions. Now, if you compare these vectors of functions, what you see is a curious observation. On the left-hand side, on the right-hand side, so over there is the relationship of the vectors of functions of the microbiota in the pre-oil, post-oil, and oil times. So the darker the square, the more similar functionality of the microbiota in a particular sample. So you see that all the pre-oil functionality seems to be similar, the post-oil functionality seems to be similar, and the oil functionality seems to be similar. But you also see the individual relationships. So the post-oil microbiota looks more like the oil microbiota. So if I was to display this on a dimensionality reduced space, what I see is that there is almost going from the right-hand OS4 empty triangle side over here, there's almost like a disruption and then a coming back in terms of the functionality that these microbes are carrying out. So there's obviously a relationship between microbiota's environment and functionality, in case you were doubting that that exists. But a very important thing is that, as I said, alignments are not an optimal way of carrying out the assignment of microbial functionality. In fact, only 2% of the reads of any given microbiome can be assigned a function, right? So we're looking only at about 2% of the functionality. What we want is 100% of the functionality. And Adrian Corfirst in my lab had built this machine learning, deep learning model of the bacterial language of life that we could then use in order to try to annotate more functionalities. And it's called Looking Glass. Now, I'm not going to the detail of Looking Glass, but the idea is that if you look at homologous and non-homologous sequences, you see that there is a difference in distributions. So each dot, if there was a dot in this box plot, would represent a pair of sequences that are homologous in blue or non-homologous in red. And you can see that the similarity between these vectors representing each of the sequences is actually higher for homologous sequences than non-homologous ones. But here's a kicker. They distribute very differently across the entire span of scores, right? So that on the right-hand side, what you see is sequence similarity, right? So obviously non-homologous sequences are not sequence similar, which is expected. This is the definition of non-homologous. But on the top graph, what you see is the distribution of homologous and non-homologous comparisons of the embeddings. And you can see that distributions are not so obviously different. So what is it that the embeddings capture? And Adrian had shown that the embeddings capture really well where the particular reads are sampled from, right? So you could see the color scheme is where the environmental package comes from. So is it from water, from soil, and so on and so forth. So this is actually a very good signature of the fact that these embeddings, rather than capturing sequence similarity, capture functional similarity. So we use these embeddings in order to try to predict functionality. And here are a few comparisons, right? So the y-axis is the observed class of the enzymes, and the x-axis is the predicted class. And what you see is that at all digits of the enzyme commission number, we're actually able to make predictions that are correct most of the time, right? So Adrian was able to identify the functionality of the reads using this data. So one thing that I want to point out for specifically oxidoreductases, what she was able to do in blue, what you're seeing is the prediction of oxidoreductases in a space where neither MyFaither or MGREST alignment-based methods are able to identify as many oxidoreductases. So she predicted lots and lots more oxidoreductases that we've never seen before. And what's important is that these predictions actually corresponded to environmental expectations. So oxygen minimum zones had significantly more oxidoreductases than did the surface, which is what was expected and was not observed for the homologous annotations. So finally, really briefly, only a couple of slides, Ariel Aptekman had built a method for predicting metal binding from sequence. So Vic had mentioned this before. And some of the interesting observations were that the predicted proteins that bind metals do not differ significantly across depth. This is the Black Sea of the Black Sea. They do not differ significantly, which is not expected, because what we were suggesting is that the environmental changes should have an impact on the proteins that are produced by these bacteria. But what you see with this black line here is the vector distance, right? So each of these protein quantities are minimal, but taken together, the differences between any two spots are sufficient. And the idea here is that the biggest difference is at the photosynthetic limit. So from a zone where there is still photosynthesis to a zone where there is none, you see a very significant drop in metal binding. And there is a thing up there, I don't know what that is, but the idea here is that you could also look at other samples gathered from different spots, and you see, based on the color schemes of these patterns, that within a particular sample set, you see similarity, but across you don't. So you can compare, let's say, soil to gut sample, and you see that they're very different, but within the gut, they're not. Okay, so back to this, the idea is to, now that we have all of these data, is to get a sense of how one maps to another. Can we go from the protein structures to the functionality? Can we go from the functionality to the environment and back? And that part is missing. And now 5 years into the project, I am a little sad that it is, but we have a new postdoc joining the lab. And he just yesterday got a visa appointment. So yay, hopefully he'll be here. Great, so that's that.